So the first of these bookish topics is depression. And a brief note about the papers, because this is the first of 3 weeks that we are going to look at papers weekly. And you might have seen the papers posted, the schedule posted, the groups posted, and I'll add to the schedule each week as we go. And the way that I've set up the discussions is as follows. So there are 2 papers each week. You will read one paper. Even groups read one paper, odd groups read another paper. And right now these are older papers, they're foundational papers, and they're meant to complement each other. So we have what we know as far as a dose response for exercise and depressive symptoms. And then does that dose response hold up over time? We want to look at a persistent effect, just of exercise and of depressive symptoms. There are many other newer studies that add to this and look at it as a comorbidity, and we might talk about some of those coming up. But only these foundational papers, and then we'll see how it goes this week, and I'll maybe change the approach for the coming weeks. But the way that I want this class to go is on Thursday, it's all group discussion. So I'm leading you through the preamble today, and Thursday, group discussions. So for the initial 15 minutes, it'll be small group review. Everyone should have read the paper coming in. You might have questions about the paper. And then in your numbered groups, 1, 2, 3, 4, you just discuss amongst yourselves. Nice small group discussions, not in front of the class, not one-on-one. You can poll each other, make sure that you understood the topic correctly, that if there's something you don't understand, ask for clarification. Small group discussions just to bring everyone up to speed. Next, I had the original plan of having what's called fishbowl. It's a teaching technique where there's one small group discussion that's observed by a second larger group. It seemed harder to pull off logistically. So what I'm doing, at least for this week, is we'll just have one-on-one group discussions. Everyone will break up into pairs. One person will have the even paper, one has the odd paper, and then you share the results of your paper in your small group of 2. Just one-on-one sharing of the paper after you've come up to speed in your small groups in the first 15 minutes. So you have a good understanding of your paper, and you can discuss in your pairs what complementary aspects are there, what things disagree, why is there disagreement. It's trying to root out, is the paper telling you what is the paper telling you what you think it's telling you? Are there any reasons to not believe it? To try to explore the critical analysis of some of the results. So one-on-one group discussions that we'll take up as a class afterwards. So we'll come to a consensus on the papers overall, or at least on the main figures. And this should take just about an hour or a little bit more. We'll see how the discussions go. And then individual sheets are handed in, plus a peer evaluation. Both of those are posted online. And the peer evaluation modifies the mark that I will assign for the summary sheet that's handed in. So every person fills out one summary sheet. Every person fills out a peer evaluation sheet for their peer. And then all those marks are accumulated for this week and then the next 2 weeks. And then the total percentage, I think, was 10% for the discussions. Is that right? Okay. And we'll see how it goes. I might do like a drop your worst score kind of thing, or we might change the one-on-ones for next week or the last week. We'll see how this week goes and go from there. Yeah, question?
I was just wondering about the summary sheets. Are those something that we complete before we come to class and before we have the discussion, or did you want those to be completed as we do the discussion?
They have to be completed before you leave. Usually it's a good idea to complete them beforehand, or you can make notes and then fill out as you go, but you're going to be distracted and focused on relaying the information in your paper to your peer, and you want to contribute to the small group discussions, so you don't want to just be recording ad nauseum when you get here. Have an idea before you get here, and then you can fill in the gaps or add to it if you want, but it has to be filled out before you go. So it might look something like this. We have a really weird number of, well, not a weird number. 14 is not a terrible number to work with. If we had 12, I could think of a great way to set this up. But we have 14 people, odd groups, even groups, and then I might just match you guys up with letters, like the A's go together, B's go together, etc. Maybe I'll change that each week. There's some room for this to evolve, we'll put it that way, but we'll come up in pairs accordingly so that each paper is represented and you can get a nice intimate discussion about the particulars of your paper. So somewhat clear? Yeah?
When do we pass in the peer evaluation?
End of class on Thursday. Yeah. You would have all the information to fill it out after reading the paper, and you need to read the paper before class. So have it filled out and then hand it in at the end of class. You can add to it if you want to on Thursday, but should have it filled out. All right then, let's talk about depression as a chronic disease. Depression as a chronic disease. And the word depression tends to get thrown around a lot, referring to various psychosocial levels of dysphoria, perhaps. It's a spectrum, and what I'm going to refer to as depression in this section is major depressive disorder. So not mild feelings of dysphoria, not feelings of malaise, but a clinical diagnosis. And I do want to acknowledge there are steps along the way before you get to a clinical diagnosis, and the information that we discuss is applicable to the clinical diagnosis, but it should also be applicable to the steps along the way or the earlier points in the spectrum. Perhaps just a lesser effect for lesser symptoms. But depression as a whole describes not just yes or no, on or off, a binary disorder. It's a spectrum like any other chronic disease. And it often occurs with other chronic diseases. So in holding up with our understanding of current non-communicable disease, it's a constellation of disorders that tends to fit together. And it might not be that depression causes coronary heart failure or obesity, but feelings related to those conditions can often contribute to or result in depressive symptoms. And so when considering a therapy or treatment, it's not often as easy as just saying energy in, energy out, or what's your insulin sensitivity. There are real people that have to execute a plan that you might lay out for them, and they might observe, or you might observe some of these symptoms in that population. So there are ways that we can accommodate that. But what I'm going to try to do is acknowledge that it does occur with many chronic diseases and then ferret out the effects of exercise on depression only. Separate them. I want to look only at depression in this section. So clinical major depressive disorder is typically diagnosed according to the Diagnostics Statistics Manual of Mental Disorders, DSM, and I think the most recent version is 5, at least of 2013 it was 5. It's possible it's been revised since then. But in the evolution from 4 to 5, not much in the diagnosis has changed. And the diagnosis is generally as follows. It has to be completed over time. 5 of the following symptoms are present during the same 2-week period leading up to diagnosis. And there needs to be a change from previous function. So in those 2 weeks, something has to have changed from baseline. And one of those factors has to be there is a chronic, constant, depressed mood most of the day or the majority of days in that 2 weeks. Now, what depressed mood means, I'll leave to a specialist, but there are definitions for what a depressed mood would be or the relative rankings of mood in a day, as long as quote-unquote depressed mood is present for most of the day. That's one of 2 possible outcomes. The other is a change or reduction in interest in all or most activities from baseline. So one of these has to occur, and then 4 other symptoms on the next page would round out the diagnosis. If neither of these is present, it's not major depressive disorder. It's not clinical. If 4 this plus 3 others are present, it's not major depressive disorder, at least by our criteria for evaluation. So 5 of the following symptoms, including one of these, at least one of these, it could be both. The other elements could be physical changes in weight or appetite, insomnia or hypersomnia, either can be present, psychomotor agitation or slowing and general malaise and lack of reaction time, for instance, feelings of fatigue or loss of energy, apathy, feelings of worthlessness or guilt. And there's no mention here about the frequency or the persistence of these emotions, just that they do occur. A diminished ability to think or concentrate if there are tangible real-world effects that might be borne out in test taking or job performance, and or recurrent thoughts of death or suicide. So 9 total possibilities. If 5 of these are evident, including at least one of the first 2, then according to the DSM manual, the individual is clinically depressed. Clinically depressed. It's also important to note that these feelings shouldn't be brought on artificially. They shouldn't be brought on by a substance. They shouldn't be brought on by bereavement or trauma. These are in the absence of some extra normal circumstance. So it's not related to another medical condition or medication or trauma. They are in and of their own, in and of their own right. And accordingly, if we use this as our clinical diagnosis, I'm actually just thinking, I don't know how they get the stat, but 80% of individuals that are diagnosed this way are diagnosed correctly. And I guess if this is the way to diagnose clinically, how do you compare that to something else? If this is the best way to diagnose, how do you know if it's right or not? I'm not exactly sure. But it seems to be fairly effective, at least by what I'll call a professional opinion, which isn't mine. But this is a rather intensive method for evaluating clinical depression. Monitoring over time and pretty significant evaluations. You need to be brutally honest in some senses. And it's not something that a lot of research studies can do if you're not in this area. So we use surrogate questionnaires in clinical type interventions to evaluate the effect of exercise or a treatment or therapy on depression. And one example of that is the Hamilton Rating Scale for Depression. It's a questionnaire you can download online. It's not exclusively the only questionnaire. There are a number of other ones. We'll look at the Beck Depression Inventory on the next slide. The CES-D is one that we see in some of the studies I'm going to refer to. The Center for Epidemiological Studies Scale of Depression. Point is, there are a number of questionnaires with questions like, do you have a depressed mood on a scale from 0 to 4, with 0 being no, 4 being the worst? Rate your score here, add the scores to each question up at the end. It's not clinical, but it could be diagnostic, and it could be used as a tool to track changes over time because it's easy to administer. But it's difficult to compare results between tests, for instance. So this versus another questionnaire. How would you compare those? Or the Hamilton Rating Scale, some questionnaires have 21 questions, others have 17. How do you compare those? Generally, the questionnaire with 17 questions is what we use as standard. And the threshold score of 8 is what diagnoses someone as having depressive symptoms. And I won't say depression, but depressive symptoms. A score over 8 indicates depressive symptoms. And so this is commonly used in research practices that we'll look at. Similar questionnaire here, the Beck Depression Inventory, very similar layout, slightly less wordy, larger number of questions, 21 to 25. Score correlates with severity, and you have a range at the end when you add up all the questions, saying this is my total score, and then I'm into whichever bracket as far as ratings of depression go.
How many questions do you set up?
21 commonly, sometimes 25.
And then like it has like kind of like a scale.
And this is not a cutoff, this is just severity, grades of severity, we'll put it that way. And we've used these questionnaires for decades to monitor depressive symptoms in exercise research. These have evolved a lot recently, and this is a new type of questionnaire that the university actually endorses, called Mood Path, which is now leveraging technology as somewhat of a memory booster or a convenience aid. You don't always have a questionnaire with you, or you can't recall exactly how often or for what majority of the day yesterday you felt good or bad or neutral. And so something like this eliminates those issues by pulling your phone, which you tend to always have with you, and asking at various times, how do you feel now? Rate yourself here, and you can see tracking progress in the back to show you mood changes over time. And this is based on a third and a different questionnaire, this one developed by the WHO, the World Health Organization. It's a 10-point scale that they're hoping will replace the HAM-D and the Beck Depression Inventory and the CES. But this is gaining a lot of traction in the research community as well. But then there's the, this is somewhat of a tangent, but then there's the other issue too of, you talk about having like the Facebook effect where you're looking at something through a screen. Are you answering correctly, or is it unbiased? Is it because it's like a technological tool? Is it something that skews the results somewhat? Still a lot of research in this area, but there are a lot of really nice inroads that allow us to make constant evaluations, maybe accurate evaluations of how depressive symptoms change. Now, whether we use one tool or the other doesn't matter. I think what's important is using the same tool. And if we use tools like this to poll the population, let's take a look at our local incidence rates versus the national average again. And what we've discussed so far is in this bottom row, mood disorder, which would be a score over 8 on the HAM-D, or a more severe rating on the Beck Depression Inventory. I don't know exactly which questionnaire was used to evaluate this here, but this is percent of individuals that responded with a mood disorder as classified by the questionnaire. And you can see, I think this is holding up with our general trend, a tendency for a higher incidence locally than provincially and at the national average, although it's not so distinct, it's not so clear-cut, there's not such a stepwise progression. Maybe owing to a reduced response rate, or the tendency for people to harbor biases associated with mental health issues and not respond necessarily correctly. But I don't have the stats. I know a lot of people, I think it's like 84% of individuals will have some degree of major depressive episode or a major depressive disorder at some point in their life. It's not reflected here, but some individuals, or most individuals, tend to go through episodes like that, so it shouldn't be something that holds a stigma necessarily. One thing we do notice is this is constantly higher in women than in men, which is odd. it's usually always reportedly higher in women than in men, and we see that that trend borne out here. Incidence rates are twice as high in women than in men at all levels. Holds across cultures, holds across countries. Is it that women are more susceptible to depressive issues? Is there a physical pathway for some reason that makes them more susceptible, or are they simply more honest in their reporting? It's hard to say. Yeah, Rosa.
Are those also including like postmortem depression for women? Like after they
Interesting. Okay, postpartum depression,
sorry.
not postmortem depression. I don't know if that's true. I don't know if that's true or not. I would expect those symptoms to be scored similarly to a major depressive episode, and so if the questionnaire was administered postpartum, it would be reflected in the numbers, but I don't know if the timing was taken into account. I'm not sure. Yeah, Kristen?
For all, where these numbers come from, would these always self-reported?
Yeah.
Yeah, so that kind of gives you a little
Yeah, all questionnaire data being self-reported
bit of, yeah.
has that possibility of intrinsic bias and underreporting. Absolutely. And that kind of speaks to the idea of, well, is it higher in women than in men because it's true and reflective, or is it an actual functional difference between sexes? Probably a bias in reporting, but I can't say for sure. Yeah, Leanne.
Could like birth control have anything to do with it? Because like women have to take tons of hormones that
Yeah,
not natural. Like
that's a fantastic point. Absolutely.
so many people are on.
Any external, and I don't want to say artificial, but artificial regulation of hormones might influence mood and add to these numbers. Absolutely, it could. Yeah, I don't know if it does specifically here, but that's a great point. Yeah. Regardless, we see it across most surveys, and if it's a representative population with representative women taking birth control, it's possible. Yeah, absolutely. Comparatively, I think it's important to bring up the individuals that not only report negative mental health outcomes, but self-report positive mental health outcomes. It also holds up that at least locally, women are in better mental health than men. And we don't see that stepwise progression as much locally, provincially, nationally, where we have worse outcomes locally. But I suppose there is a predisposition for men locally to have an abnormally low rating of good or excellent mental health for whatever reason. That's one number that seems to stand out. And then these numbers don't add up, which is another thing that seems to be a little bit off. Why wouldn't the numbers add up for mood disorder plus good or excellent mental health? And I suppose the answer to that is we're not really measuring the middle ground. We're not measuring people that are so-so or average or don't feel either a disorder or no disorder. So it could be that that's missing. But interesting to point this out, I think. Nice to see, however, that perceived stress is a lot lower locally than the national average. We have a nice, easy, laid-back lifestyle. This should be the East Coast lifestyle row, I think.
Put that on a shirt.
Yeah. So there is a the prevalence of mood disorders is persistent across provincial and national realms, and so there is some reason to investigate therapies. Obviously, there are reasons to investigate therapies for mental health. And so in trying to understand the therapies, we need to figure out what goes wrong, to the best of our knowledge, what goes wrong. And I'm going to try to lay out some of the tangible things that go wrong, some of the markers that we can measure that go wrong. There are other cognitive biases that cognitive behavioral therapy will address that also go wrong, that are not things that can be easily measured, that aren't included here. These are not an exhaustive list, but they are things that typically can be measured and might be therapeutic targets, physical therapeutic targets. So there is no one reason that we've pinpointed so far for what causes depression, but there are a couple key candidate factors. And some of those are listed here. These are neurotransmitters, specifically monoamine neurotransmitters that are involved in reward, norepinephrine, serotonin, and dopamine. I'll talk about each of those on the next slide briefly. But these act in the brain to form habits and trigger averse reactions to different stimuli, to encourage you to chase after others. And they also inform what we call the HPA axis, the hypothalamic pituitary adrenal axis, which is essentially a connection between mental and emotional states and the physical release of hormones, the physical circulating hormones in the body. This axis can connect those two realms, a mind-body connection, for a lack of a better term. And we see whole body effects in depressive individuals. They have higher levels of inflammation, higher levels of stress and cortisol. The hormones involved in this axis are dysregulated. They are apparent in different levels. So we observe a physical manifestation of what we would consider a cognitive disorder, which really kind of suggests there's a lot more that we've yet to learn. That it only really scratches the surface, that we're starting to acknowledge that there is this physical connection between emotional and then whole body effects. Autonomic nervous system dysregulation you can see in heart rate variability. Heart rate we measure over the course of a minute and we get one number, but every cardiac cycle expands and contracts slightly. And how it expands and contracts is variability. And a lot of people use that number for training, and specifically overtraining. If it's inflexible and static, it's a sign that you've overtrained. If it's fluid and flexible, it's a sign that your heart is healthy. And so this autonomic nervous system dysregulation, you can see borne out in measurements like that as well. There's platelets and endothelial reactivity, a whole bunch of measurements that we can see are borne out with major depressive disorders. But the connections between the whole body manifestation and the mental or cognitive dysphoria are where we would apply our therapy. Otherwise, you're just treating the symptoms. So if you want to treat the root cause, you'd want to interrupt the thing that goes wrong, or the thing that connects the cognitive disorder from the whole body manifestation. And to the best of our knowledge, or the best of our experience so far, it seems that these neurotransmitters have some role. So norepinephrine is the main messenger for the sympathetic nervous system, the thing that helps to turn on the muscles, turn up the heart, constrict the vasculature when you move to exercise. It's a common neurotransmitter, tends to enhance vigor, alertness, perception, acuity, visual and mental auditory acuity. It makes individuals anxious and restless if it's persistent in high amounts chronically. You get that jittery feeling. And so dysregulation of norepinephrine could have inroads into affecting the HPA axis and resulting in physical manifestation of depressive symptoms. And it is a metabolite of dopamine. So dopamine is the precursor for norepinephrine. And dopamine itself is a neurotransmitter, and that's the classic reward neurotransmitter. You feel a sense of accomplishment, or you feel happy to have submitted a paper on time or succeeded in your job. It's dopamine that brings about those feelings of reward and reinforces those reward pathways. And a lot of addictive substances leverage those reward pathways to cause addiction and have you come back to them. Now, reward pathways all in the head. That feeling that feeling of reward is all central, but dopamine does have a role in improving insulin sensitivity peripherally. There's some suggestion it helps intestinal motility. So there are other roles in the body other than just a neural reward pathway, but this is how we interpret it in the context of depression. And then lastly, serotonin, it's not necessarily reward hormone, but it helps to regulate mood, appetite, sleep, happiness, feelings of happiness. So more serotonin in circulation is related to more feelings of happiness and connection with people and things around you. And MDMA, psilocybin, LSD all stimulate serotonin release to leverage that or increase that feeling of connectedness with other individuals and those feelings of happiness. And so dysregulation of serotonin, if you have too much uptake or breakdown, you have the opposite that occurs, too little happiness. And if that persists for a long period of time, you can see how that might affect feelings of depression as well. This is the neurotransmitter that was mentioned in that video that we watched that was produced in the gut. 90% is produced in the gut. And that might send signals through the vagus nerve to the brain and help to reinforce happiness and mood and cravings that way. Or it's circulated through the blood back to the brain where it exerts its effects. But really interesting to note that 90% of the body's serotonin is produced in the GI tract and not in the brain proper. So collectively, these are what we call monoamine neurotransmitters. They have one amine group. They're similar derivatives, have a similar function to modify mood. I do want to bring up, I don't have this written on here, but I do want to bring up while we talk about serotonin, one popular class of drugs, medications to treat depression are selective serotonin reuptake inhibitors, or SSRIs. And what they do is prevent serotonin that's free from being stored away. They prevent the feelings of happiness associated with serotonin from being removed. And so one of the most common is commonly known as Zoloft, or we'll see it as sertraline in the research. And we often compare sertraline to exercise or sertraline to a placebo. And that is acting to prevent the reuptake of serotonin. So it's increasing the circulating concentration of serotonin. There are other types of antidepressants as well that prevent breakdown of serotonin and other things, but I won't go into detail about them. Now I want to introduce this slide, and it looks messy, and there's a lot of detail, and it's true, there is a lot of detail, but you remember from second year anatomy and physiology, this is a sagittal view of the brainstem, the pons here, the midbrain, the thalamus. This is the third ventricle where the cerebrospinal fluid is. You've got the fourth ventricle down here. The whole cortex is off the slide. And this is what we call it's the midbrain or subcortical region, which you might think of as the reptile brain or the monkey brain, the visceral brain, where we respond to emotions. And these are things below our awareness. There are automatic responses, fight or flight, etc., that are really born out here. And the point of bringing this up is not for you to know the hypothalamus and the pituitary gland and the hippocampus and the ventral tegmental area specifically, but just to acknowledge all of the pathways that have been identified between these subcortical regions and how they might regulate mood or how they might regulate signals between these subcortical regions and then the prefrontal cortex. So we have some structures that are involved in reward, fear, motivation, and there are signals between them. And some signals are good, some signals are bad. Dopamine is, there's dopaminergic input according to these blue lines. You can be aware of feelings of reward if that is elevated to the prefrontal cortex or to your conscious awareness. But there are many different overlapping signal pathways for dopamine, as well as for norepinephrine and serotonin. Serotonin is also known as 5-HT, as well as some other typical neurotransmitters. The point is there's a lot of overlap and redundancy and places for things to go wrong. I'll put it that way. There's a lot of places for things to go wrong. I used to go into a lot more detail about what all of these structures did and what types of emotions they were responsible for, but I don't think that's terribly important. What I want you to take away from this is that there are inputs for our typical monoamine neurotransmitters to the hypothalamus. That's basically what I want you to take away from this. Because the hypothalamus is central to that HPA axis, the hypothalamic pituitary adrenal axis. And it's at the center of monoamine regulation in the brain. And really that's what this is showing.
You said it was
Yeah.
at the center of monoamine regulation in the brain?
It is it's subject to regulation by serotonin, dopamine, norepinephrine. I'm trying to build the connections between neurotransmitters and then the whole body manifestation of depressive symptoms. So if we take the hypothalamus as being central in this subcortical region and in regulating things like blood pressure, heart rate, stress, cortisol, fight or flight, which it does, we can see how it might be the relay between those monoamine neurotransmitters and a physical endocrine response. So the paraventricular nucleus is a part of the hypothalamus. And it's one specific part that we've identified initiates or is largely responsible for initiating the HPA axis or activating the HPA axis. So we're drawing our scope outwards a little bit. We're moving out from the subcortical region only. We're simplifying the HPA axis here. You can see the pituitary, the adrenal gland, the kidneys, and the stepwise release of hormones. Their names aren't necessarily important, but if you want to know them, corticotropin releasing hormone is released by the hypothalamus and activates the pituitary release of adrenocorticotropic hormone, which then at the kidneys releases glucocorticoids to prompt a stress response, a fight or flight response. So we have the hypothalamus initiating the response, and it is subject to regulation by monoamine neurotransmitters, by the amygdala. You can see a positive stimulatory relationship there, or it's inhibited by the hippocampus. And those other regions are also subject to regulation by serotonin, dopamine, norepinephrine. If the HPA axis is activated, typically the negative feedback loop will turn it off. If this is an acute singular response, a negative feedback loop exists where once the stress response has circulated and exerted its effects, it will return that signal to the brain to specifically decrease the activity of the hypothalamus. It turns off the HPA axis. So there should be acute regulation whereby this is short-lived.
Can you repeat that? If what is activated?
If this stress response, if the HPA axis, which initiates the stress response, is turned on, after that signal has been carried through the body to do whatever it's supposed to do, it should return back to deactivate the hypothalamus. Classic negative feedback loop. If something is, if blood pressure is too high, that signal is interpreted by the brain to reduce blood pressure, to reduce heart rate, to vasodilate, to return the signal to normal. Negative feedback loop. Same thing here. If glucocorticoids, which is a form of stress marker, are active and present in the circulation, they will turn off the hypothalamus to inactivate this axis, the stress response. So normally, this is acutely regulated within the bounds of that negative feedback loop. But if there's ever chronic activation, and we don't know how that happens, but in major depressive disorders, that does occur, we observe it. If this is chronically activated, and we have persistent high levels, specifically of glucocorticoids in this case, they can actually damage areas in the hippocampus, causing it to atrophy. And we measure that atrophy in depressed patients. Using MRIs, we can see that the hippocampus is smaller. And so damage to the hippocampus removes the inhibition on the hypothalamus. It takes the brakes off, allowing this cycle to propagate, allowing chronic activation of this stress response. And this could be the manifestation of dysregulation in neurotransmitters and the start of a physical consequence, a physical constant chronic stress response.
So it's kind of like the hippocampus can't close
Yes,
the negative feedback loop. Basically.
that's right.
Yeah.
Yeah, exactly.
And then, so would you see any damage to the kidneys because the adrenal cortex are constantly firing?
Good question. Is there damage to the kidneys? I don't know if the kidneys themselves would be damaged. If there would be damage, it would be the gland. But I think the gland is supposed to respond by releasing hormones. I don't know. It depends on the supply or if there is a decreased sensitivity. I don't know the answer to that. I think since we observe persistent high cortisol and glucocorticoids and atrophy of the hippocampus, I'm going to say no, unless there's some other reason that those responses would exist. But right now, as far as we understand it, this is the main way that that plays out. And so I'm going to say no, the adrenal cortex itself is not specifically damaged. Yeah, it does.
How do the glucocorticoids damage the hippocampus?
I don't know. We just know that it does. We observe a high circulating level that correlates with a decrease in hippocampal volume. Razan first. Yeah.
I'm confused with why the HPA axis is bad. Because I thought it was supposed to connect like the body and the mind.
Yeah.
Right? It's like,
It's not bad, but in a situation where it's always active and can't be turned off, that brings about low-grade chronic stress and inflammation. That might be how persistent depressive thoughts result in a physical effect. Chronic activation.
so
Turning it on is not bad if you can turn it off.
function of the HPA. So like what exactly does it do? Because I thought it's a positive thing that it's connecting with the body and mind.
It's a positive thing that I think the best example might be a fight or flight response or a stress response, something that prompts you to act. This is a stress response, and normally when the stress is absent, the axis will be turned off or won't be turned on. But if there's ever chronic low-grade stress that always turns this axis on, and we have persistent high levels of glucocorticoids, we get damage that prevents normal functioning of the axis. It's kind of like how we get high blood pressure. Blood pressure should be regulated around 120/80. But if we're always insulting it and always raising blood pressure, we lose the ability to regulate back to normal. Same thing here.
So what kind of stress would activate the HPA? Is it like different thoughts? Or
It could be fear. It could be, I think modern day stresses might be like exam taking or something that requires like scrutiny in a public forum, for instance, like any kind of low grade chronic stress like that. That's my understanding. Dylan?
As far as the glucocorticoids, do they have any effect anywhere else in the body that you would be able to regulate?
Good question. Possibly, and I don't know them because I'm only thinking about them in this context.
Okay.
Yeah.
I'm just thinking, like, would you see like an increase in the heart rate variability or something like that? Not sure.
Great idea. I don't know. I really don't. Yeah, Peter?
Would a possible treatment for depression be like a way of reducing the amount of glucocorticoids?
Yeah, I think so. It could be. That's if it doesn't. So there's also possible negative side effects. If you inactivate or decrease circulating glucocorticoids, do you lose this axis? And are there other things that this response is doing other than this stress response, which we've highlighted here? And would losing those things be bad? I'm not sure. By this simple schematic, if we can get rid of glucocorticoids, we could fix the problem. But I think there's probably other effects that we're not looking at here that might complicate that response. I'm not sure. Great idea, though. One thing that we do know, and we've connected this stress response to physical changes in the brain, one through the atrophy of the hippocampus, but also through something that we call neurotrophic factors, which is a brand new, within the past 5-10 years, area of research. And the candidate neurotrophic factor is this guy, BDNF, brain-derived neurotrophic factor. And BDNF is one class of I'm trying to think, is it best described as a transcription factor or a protein or an enzyme? Hey, let's call it neurotrophic factor. It's one class of neurotrophic factor. There are others, and their job is to regulate branching within the brain. And so you can imagine if we disrupt this and it acts at the hippocampus, maybe we can get decreased branching and atrophy that we observe on MRIs. This was the first one to be identified and characterized, but there are many others. It still remains like the typical neurotrophic factor that we look at with exercise or with other stresses. And so in a normal situation, you can see BDNF here, which at the dendrites helps to regulate branching. And this is a normal state with normal branching. And this is an example of a pyramidal neuron, which is just the typical neuron in the prefrontal cortex. It's not the only type of neuron, but it's just an example of branching in a specific type. So normally, BDNF allows that branching. And not all of the dendritic locations are branched, but that's fine. We don't need that. Importantly, this is modified by monoamines, neurotransmitters, modified by glutamate, which is an amino acid, modified by others, which we'll see in a second, so that if these are present, it affects how BDNF is produced. They affect how BDNF is produced. And they affect how specifically it's transcribed. So CREB is a cyclic AMP response element binding protein. If you remember way back to genetics, it's a thing that binds at the start of a gene and turns on the transcription of that gene. Specifically here, the gene is for BDNF. It affects how the DNA for BDNF is produced or transcribed. These things affect the release of BDNF. In a normal situation, with normal levels of monoamines and glutamate, we have some level of branching. In a depressed scenario, Other factors that modify BDNF release are persistent high circulating glucocorticoids. They inhibit the activation of transcription of BDNF. What does that mean? That means less BDNF is produced. There's less BDNF to regulate branching. And you can see what we call reduced arborizations, reduced branches in the dendrites of those neurons. Reduced branching, which is a precursor to atrophy in some regions of the brain. So now we're starting to see, okay, if there's dysregulation of the neurotransmitters, we can activate the HPA axis. Chronic activation means there's many or a high circulating concentration of glucocorticoids. Not only do we have those that disrupt BDNF function, but we've already acknowledged there's a dysregulation of monoamines. We don't know what effect that has. Overall, less BDNF is produced. We see less branching, which is indicative of a reduced function, reduced volume in those regions of the brain. Yeah, Leanne?
So it's glucocorticoids that inhibit the transcription of BDNF.
Yes. Yeah.
Only in surplus amounts though, correct?
I don't want to say only in surplus amounts. They'll always inhibit the transcription of BDNF, but normally glucocorticoids aren't present in a high enough concentration or for long enough periods to have a significant influence on transcription. They'll always inhibit it, but only when there's persistently high levels will that culminate in a noticeable reduction in BDNF. Let's put it that way. And on the flip side, if we can ever increase BDNF release, which happens in a treated state, we can recover that branching and even increase branching in some neurons beyond what would be considered normal. BDNF release is something that we observe with almost all antidepressant medication administrations. We observe it's released with exercise. One study that we looked at a few years ago did jugular artery and vein blood samples and measured the difference in BDNF across the jugular artery and vein. Well, the only thing between the jugular artery and vein is the brain. We don't know where in the brain, but the brain produced BDNF in response to exercise, which is a really nice way to lead into our next discussion on how exercise might impact depressive symptoms. It might do it through recovery of BDNF transcription and increased arborization. Yeah.
So the schematic, it's saying that increasing BDNF can also increase the production of BDNF.
Yeah, BDNF is a, what's the word? Self-coactivator? No, that's not it. BDNF magnifies its own production. That's absolutely true. I forget the term that we use for it, but it's also seen with PGC1 alpha, which is a transcriptional coactivator in the mitochondria. I can't remember it. Yes, it does. It is a positive feedback loop, but there's another name for it that I'm forgetting currently. But the idea is the same. Positive feedback loop, it will magnify its own production. Yes. What this isn't telling us is this the reason for depressive symptoms? Is the level of arborization the reason for a change in depressive symptoms? Or is this simply another symptom of those upstream depressive symptoms? We don't know. We're not exactly sure. We know BDNF release changes with exercise, and it's inversely proportional to the appearance of depressive symptoms, but we're not sure which is causative. But we do observe at the whole body level Exercise, yes, it increases BDNF release, so it should increase arborization. It should help to limit the negative effects of the HPA axis and chronic glucocorticoids. We also see that depressive symptoms, as scored by a HAM-D or another questionnaire, are lower. Symptoms are generally minimized with greater fitness, greater activity, and it has similar effectiveness to medication. Okay, good. Yeah, I thought I had worded this incorrectly. It has similar effectiveness to medication, but it's more effective in follow-up. Remission rates longer term are higher with exercise. That means the effects of exercise seem to last longer. This is, I meant to take this out. This is an artifact from when we used to do a lot of prescription in this course. And this is meant to refer to the idea that with depressive symptoms, when interpreting data at a high intensity, or when interpreting results of a VO2 max test or a graded exercise test, they have to be taken with a grain of salt, because for those types of high intensity tests, motivation is a key factor. And in a lot of individuals with or exhibiting these symptoms, motivation is an issue. And so the physical tangible results of a VO2 max test may or may not be compromised. But the heart rate response at a given exercise intensity is not something you can fake. It's how far you can go during a graded exercise test that might be altered. So let's just leave that for now. Ignore that for the time being. But I do want to bring up or dive into these top 2 points. We want to talk about how exercise influences symptoms. If we have a rudimentary understanding of the pathophysiology, that's great. We can look at how exercise affects those individual neurotransmitters, but really what we care about is at the whole brain level, are depressive symptoms reduced? And absolutely, across the board. And this is a return to the aerobic center longitudinal study, wide epidemiological study across a broad swath of the population. 100,000 subjects, I think, at this point in the early 2000s. What we're looking at here is fitness. So always lower depression scores. This is the CES-D, Center for Epidemiological Study of Depression. Like the HAM-D or the Beck Depression Inventory, a questionnaire that evaluates depressive symptoms. Lower scores with higher fitness. Stepwise reduction in scores with higher fitness. Awesome, great. Now, did the fact that you have a higher fitness decrease those scores? Or is it that you are less depressed that allows you to exercise and gain a higher fitness? Not sure. But it's pretty nice that these agree and there's a stepwise correlation with higher fitness and lower depressive symptoms. Also notice higher in women than in men. Again, across the board, translates into almost all observations of depressive scores. What about not physical fitness, but what about physical activity? Do you need to be fit? Do you need to be an elite athlete to reap these benefits or observe this correlation? The answer seems to be no. If you have enough physical activity over the course of a day, you get the maximum effect insofar as reducing depressive symptoms. And sufficient activity, in this case, sufficiently active, when you dive into the paper, is essentially 10,000 steps per day, 150 minutes per week of moderate intensity exercise. So it's the guidelines, the CSEP guidelines that we've talked about at the start of the course. If you meet those guidelines, you reap the maximum benefit, apparently, of reducing depressive symptoms. Now again, we're not sure, is this a result of being physically active, or does being less depressed allow you to go out and be more active in the wide world? We're not exactly sure. But these both agree. Higher physical activity, higher fitness is associated with lower depressive symptoms. Again, higher in women than in men. So what we want to see is an intervention. We want to induce exercise and monitor a change in depressive scores. And while we're at it, why don't we compare that to medication and to a placebo? This is 4 months of either exercise at home, what the normal average person in the population would do, supervised exercise in the gym, so you're not cheating, medication, and I forget the dose, but let's say doctor-approved medication, versus a placebo, sugar pill. 4 months, matched exercise, 30 minutes a day, 3 days a week, moderate to high intensity, 70 to 85% of heart rate reserve, which is roughly equivalent to VO2 reserve. And the medication, again, sertraline or Zoloft, which we identified as an SSRI. These are HAM-D scores, questionnaire scores, evaluating depressive symptoms at the end of 4 months. And this graph alone doesn't tell us a whole lot. It tells us at the end of 4 months, HAM-D scores are similar in all groups. It doesn't tell us anything about how they changed until you look at the original scores, which I've overlaid here, 17, 16, 16, 17. Really high. This was a 21 question HAM-D. Was it? Yes, it was. 21 question HAM-D, very high. Major depressive disorder. I would wager to say clinically depressed individuals. Very high original scores. After 4 months, decrease. Excellent. Exercise decreased HAM-D scores. Not only that, exercise at home decreased HAM-D scores. Maybe they were as effective at doing the same exercise as the individuals that were supervised. Maybe not. Free living individuals on an exercise program saw just as much reduction as those that were supervised. And this is as good as medication. Exercise here is as potent as medication over 4 months in decreasing HAM-D scores. That in and of itself is a fantastic finding. But none of those are the coolest finding. What's the coolest finding? Placebo. Placebo, as effective as medication, as effective as exercise. Now that's not to undermine the concept of major depressive disorder. It's to suggest that perhaps the therapy has a lot to do with your expectations, right? And if you expect you're receiving treatment, maybe you're hopeful, and maybe that hope helps to provide some kind of mental boost. I don't know, that's not my area, but that placebo has a measurable effect that's in line with the rest of these groups is pretty striking. I'm not going to leave it there, though, because I don't want you to take away from this that, oh, placebo's good enough. I don't have to exercise. It's important to note none of these groups, on average, are in remission. For the HAM-D, the cutoff is a score of 8. A score under 8 means no depressive symptoms. Score above 8 means still some mild cognitive dysphoria. None of them are completely remissed. But on an individual basis, when looking at remission, those individuals that were in remission, higher in exercise versus placebo groups. And medication, which has a very similar percentage to placebo. Half of the individuals were in remission at 1 year post-trial with exercise versus the other groups. So when evaluated 1 year later, 50%, 45 or 50% of the individuals with exercise had a score lower than 8. 30% in the placebo and medication groups had a score lower than 8. Still pretty good. Some treatment worked. Exercise seemed to be more effective in remission. All right, we got 5 minutes. I just want to touch on this other quick study before we get into our information. Before we get into our papers on Thursday. Our understanding of the monoamines, neurotrophic factors, Zoloft or sertraline, placebos, isn't all-encompassing. There's even some suggestion that oxidative damage can play a role. Oxidative damage seems to be popping up everywhere. We saw it in mitochondrial dysfunction section of diabetes, free radicals, reactive oxygen species. It seems there might be a role for depressive symptoms or for free radicals to influence depressive symptoms as well. And so this is in rats that were injected with BSO, and it's got a long, complicated name, and I'm not going to tell you what it is. All you need to know is that it stimulates oxidant damage. When you are injected with BSO, it inhibits the body's defense mechanism, and so you can't defend against the oxidants that are normally produced. So they wreak havoc. This is control versus 4 weeks of either exercise or not, with oxidant damage or not. In rats, so we have a lot of control over how we administer it. And this is an attempt to measure depressive symptoms by evaluating the percent of time in the center of their cage. The idea being that if you're depressed, you'll be reclusive or hide away as a rat. If you're not, you'll spend more time in the middle, hanging out with other rats. You can't ask a rat to do a questionnaire, so this is the next best thing. And we see a pretty nice, decrease in the time spent in the middle with oxidant damage. So the oxidant damage did something to make them stay away. And this was recovered completely with exercise. Even with the oxidant damage, exercise protected against whatever the negative effects were, which is pretty cool. But you could argue, okay, time spent in the center of the cage might not be the best measure of sociability. What about nature's best marker of sociability? The number of rearings, or the number of pups, or the number of litters, I'm not sure. Anyways, higher number means higher number of rearings, more pups being born to those individuals or those rats. Lots of sociability in all groups except those with oxidant damage. Maybe there's some defect in the machinery, who knows? Maybe it's depressive symptoms, but completely protected against or recovered with exercise. And if you dive deeply into this paper, this is looking at their behavior, but this mimics changes in the urine, the blood, the muscle. The oxidant damage itself was prevented in the exercise group. So even with this BSO that stimulated the oxidant damage, exercise recovered it, exercise prevented it. And so if this is an appropriate marker of depressive symptoms, this could be one of the methods by which exercise works. Let's just pop over to the conclusions quickly. You have two other slides which basically reiterate what we've talked about already, the proposed mechanisms of exercise, monoamine theory, neurotrophic factors, oxidation. But let's just evaluate the conclusions because you don't have these points. And I'll just say there are many treatments that work. We've looked at a few of them today that are promising. But there's a lot outside of the scope of this lecture that do work effectively. Cognitive behavioral therapy. Medication works, even though I'm trying to convince you exercise is as good, if not better. And certainly exercise works as well. The exercise that seems to be most effective, or that we have data for that is effective, is moderate aerobic exercise. It reduces depression scores. The higher your fitness, the higher your physical activity, and if you engage in exercise, the lower your reported levels of depression. We don't know how or why, but we know that it works. If it's because of the monoamine theory, is it because of neurotrophic factors, or is it because of oxidant damage? We don't really know, but we know that it works. And I just want to reiterate, I think it's really cool that there's such a tangible, potent effect, even in placebos. It really taps, or it points to that we don't really understand, one, how this condition manifests. We don't know the best way to treat. We've got some good candidates, but there are many other things at play that are question marks, are mysteries at this point. We can't account for why a placebo works with exercise or in comparison to exercise or medication. It's an unknown, at least from our point of view. So I'm going to leave it there. When we get to class on Thursday, we'll jump right into papers. They are dose response and then the longer-term remission of exercise in depression. We'll see how the discussion goes on Thursday. We'll retool if we need to. Question before we go?
Just what was CBT again?
Cognitive behavioral therapy. It's therapy we haven't talked about, but it's very effective from a psychological point of view. Have a wonderful Tuesday. And yeah, see you Thursday.
For the group papers, is it based off on lab groups?
It's all Moodle.